I will say, for the record, that I signed the consent documentation willingly, read every clause, and had no objections to any of it. The Tharon Institute's legal framework was thorough. 14 pages in a script I cannot read, rendered into my language by a translation engine that got most of the idioms right, and one of the Institute's junior researchers, a small, four-limbed being named Vethurin, sat across from me for 2 hours going through it section by section, patient and precise. I was the first human volunteer to enter a Tharon suppression trial. They wanted to make sure I understood. I understood. I signed. What I did not tell them, because it did not occur to me that it was relevant, was that I had been sleeping badly for 11 months. Fieldwork in deep station environments does that. The recycled air, the artificial light cycles, the pressure variance in the berthing quarters, none of it ever feels quite like sleep. I had been managing on 5 hours a night, sometimes less, and I had stopped noticing. That detail would have been relevant. I mention it now so the record is complete. The trial chamber on Research Station Orvith 9 was clean and cold in the way that all Tharon workspaces are clean and cold. Surfaces that looked like polished stone but were not stone, lighting that came from everywhere and cast no shadows, instrument arrays built into the walls at heights calibrated for species larger than me. I sat on the suppression platform, a flat table with a recessed headrest that did not quite fit the human skull profile, and Vethurin attached the monitoring leads to my scalp and temples while 2 senior researchers, whose names I cannot reproduce phonetically in this document, reviewed calibration readings from behind a glass partition. There were 4 technicians in the room. Everyone wore the same expression, which I have since learned to read as professional attentiveness edged with mild excitement. This was a significant trial. I was a novel subject. Vethurin explained, again, what the suppression array would do. It broadcasts a modulated signal through the neural interface leads, tuned to the passive receptive frequency of the target species sleep architecture. In every species the Institute had tested, 43, at the time of my trial, the sleeping brain presents a window. A state of reduced self-generated signal, lowered internal noise, a quieting of the consciousness that the array can enter through and seed with its override pattern. The override is not painful, not invasive in any surgical sense. It is, Vethurin said, like pouring water into a vessel that is already open. The subject sleeps. The array writes. In the morning, the subject wakes compliant, calm, and the behavioral modification persists for between 6 and 14 standard days depending on species. I asked what the array would write into me, specifically. Vethurin said, general calm, reduced threat response, a mild cooperative orientation toward Institute staff. Standard trial parameters. I said that sounded fine and lay back. They began at the lowest calibration. I felt nothing, a faint warmth at the temples, less noticeable than a pair of headphones. The instrument arrays behind the partition began producing readings. I know what those readings showed because I obtained a copy of the trial record afterward, and because Dr. Vethurin was, ultimately, both honest and generous in the post-trial debrief. The array was broadcasting. My brain was receiving the signal. And my brain was, simultaneously, producing its own signal at a volume that made the suppression broadcast look like a whisper in a room where someone was already playing music at full power. This is the part that requires some explanation, which I will try to give in plain terms, because the Tharon neuroscientists spent 3 days after the trial trying to explain it to themselves and produced 42 pages of internal notation that I also obtained and mostly could not follow. Human sleep is not quiet. That is the core of it. When a human sleeps, even a light sleep, even the thin, unsatisfying sleep of 11 months of bad station rest, the brain does not open a window. It opens a second room and locks the first one. The self-generated signal does not diminish. In most species it drops to what the Tharon index classifies as resting baseline, a low, receptive hum, an open architecture waiting for environmental input. In humans, the signal shifts register. It does not go quiet. It goes inward and generates its own content with roughly the same intensity, in many cases greater intensity, than the waking state. Humans dream actively. Humans process, construct, replay, and create during sleep at neurological volumes that, to an instrument array built for 43 other species, look indistinguishable from wakefulness. The suppression array had no entry point. Not because I was resisting. Not because I was trained against it, or medicated, or in any way armored. Because the architecture the array was designed to exploit, the passive window, the open vessel, does not exist in the human brain. There is no off switch. There is no quiescent state to enter. The array was broadcasting its override pattern into a brain that was already, at every moment, broadcasting its own. It took them 40 minutes to notice something was wrong. I was still awake at that point. Not deliberately. I was simply lying in a dark, quiet room with nothing to do, and I was tired, and my body was deciding, in its own time, whether to sleep. The lead researcher sent a query through the intercom. Was I experiencing any unusual sensations? I said no, and that I was feeling somewhat drowsy. There was a pause. They increased the array output by 30%. I know, from the trial record, what was happening on their side of the partition during those 40 minutes. The primary display was showing a neural output curve that should, by every prior dataset, have been declining. In all 43 prior species, the array's passive frequency broadcast induces measurable reduction in self-generated neural signal within the first 8 to 12 minutes of low calibration output. The subject quiets. The brain slows toward its resting receptive state. The curve goes down. The array signal fills the space the subject's own signal has vacated, and the override takes hold. My curve did not go down. It held steady for the first 20 minutes, which the researchers initially attributed to wakefulness. I had not yet fallen asleep, and a waking brain in any species will sustain its own signal above resting baseline. Expected. Unremarkable. They waited. At 30 minutes my curve had still not declined, and the two senior researchers behind the partition were, the record notes, beginning to log queries about instrument calibration. At 38 minutes one of the technicians recalibrated the monitoring leads. The readings did not change. The curve held. I was awake, lying quietly in a dark room, and my brain was simply running, full and present, without any observable response to a signal that should, by that point, have already begun to reshape it. I fell asleep approximately 12 minutes later. Not because of the array. Because I was tired, the room was dark, and there was nothing keeping me awake. This is, as I understand it, the precise moment that the trial record becomes difficult for the Institute to categorize. My sleep signal did not decrease when I lost consciousness. It reorganized, intensified in certain frequency bands, and expanded. The suppression override, which by that point had been boosted to the highest calibration the array could sustain without tissue risk in smaller species, was running directly against a self-generated dream state operating at what Dr. Veth Uren later described, with evident difficulty, as a volume they had no prior scale for. Not a pathological volume. Not a signal produced by distress or neurological anomaly. Just a healthy human brain asleep, running its interior world at full production, completely indifferent to the external signal trying to ride over it. I know, because the record tells me, that the first containment escalation was triggered 23 minutes after I fell asleep. The technicians had noted that the array was producing no behavioral effect on the subject, and that the subject's neural output was increasing rather than decreasing with array intensity. Standard protocol for anomalous trial response is to escalate to containment observation status and notify the senior researcher on duty. This was done. I slept through it. The second escalation came 40 minutes later, when a third senior researcher was called in and the team attempted to gently wake me by raising the room lights and activating a mild auditory tone. I did not wake. This is not unusual for a human who has accumulated 11 months of poor sleep and found, suddenly, a dark, quiet room. I was simply very asleep. The auditory tone was something I incorporated into a dream about a coastal monitoring station, which I remember with reasonable clarity and which was, as far as I am concerned, the best sleep I had gotten in nearly a year. The third escalation involved Veth Uren physically entering the chamber and placing a hand on my shoulder, at which point I woke immediately, said yes, sorry, is it time? And sat up. The room, I will say, had a particular quality of silence. The kind of silence that happens when a number of people have been concerned about something for a substantial period and have just seen it resolve in the most ordinary way possible, and are not yet certain how to reclassify what they were concerned about. I asked if the trial was complete. Veth Uren said, with what I can only describe as careful professional composure, that yes, the trial was complete for today, and would I please remain in the station for the post-trial observation period. I said of course. I asked if there was somewhere I could get tea. There was a debrief the following morning, then another that afternoon, then a formal session 3 days later with 2 additional researchers from the Institute's parent facility who had been forwarded the trial data. I answered their questions as fully as I could. They were good questions, precise and technically detailed, and I did my best to explain what I know about human sleep architecture in terms that translated across the language gap. The central difficulty they kept returning to was the concept of an active dream state, a sleep that generates rather than receives. In every species they had catalogued, sleep was a reduction, a lowering of the self, an opening. The idea that human sleep is, neurologically speaking, a continuation of internal activity by other means, that we do not turn down during sleep, we turn inward, required, I am told, a formal revision of 3 categories in the Institute's Sentient Species Neural Classification Index. One of the visiting researchers asked me, with what I think was genuine philosophical difficulty, what we dream about. I said it varies. A monitoring station, in my case, on that particular evening. Sometimes work problems. Sometimes nothing that resolves into a narrative at all. Just sensation, movement, the feeling of being in a place that doesn't have a name. The researcher noted this into the record and asked whether we controlled the content deliberately. I said no, not usually, though some people practice techniques to try. The researcher was quiet for a moment, then asked, so every night, without control, without intention, the interior world simply runs? I said yes. Every night. Since before we had language to describe it. Since before we had any category for what it was. Another silence. The researcher noted something else into the record and moved to the next question. I am not certain I understand why this was so alarming. I sleep. Everyone sleeps. The fact that human sleep happens to be loud enough, internally, to completely drown a signal that pacifies 43 other species is, and I want to be precise here, not something we ever designed. It is not a defense we developed, or a practice we cultivated, or a discipline we train. It is simply how the brain works when a human lies down and closes their eyes. We go somewhere. The somewhere is ours. It does not have a door you can open from outside, because the door was never built facing outward. I submitted this report to the Institute's Trial Records Office 11 days after the session concluded. I understand there is a follow-up study proposed, examining the sleep architecture of a broader human cohort under controlled rest conditions rather than under suppression. I have volunteered for that as well. It sounds, frankly, like an excellent opportunity to get some sleep. For what it is worth, I hold no reservation about the Institute or its researchers. Vethurin was professional, thorough, and genuinely curious in the way that good scientists are genuinely curious. Not alarmed, not threatened, but interested in the gap between what the model predicted and what the data showed. That gap, in this case, was the entire premise of the suppression array. I cannot help that. I can only describe it honestly and let the classification revision stand on its own. The array was built for a window that does not exist in us. We were not told the window was supposed to be there. Nobody consulted us when the index was written. We were simply sleeping the way we have always slept, building our interior worlds every night without knowing that other species considered sleep a form of surrender, a moment of openness, a vulnerability they managed and protected. We do not experience it that way. We never have. I filed this report, poured the last of my tea, and slept for 9 hours without interruption. The monitoring equipment the Institute had asked me to wear for the post-trial observation period logged all of it. I am told the data was, for the researchers reviewing it the following morning, both entirely expected and somehow still remarkable. I am not sure what else to tell them. That is what sleep is, for us. That is all it has ever been.